Hello, and welcome to the Drug Discovery World podcast, a podcast covering topics around drug discovery and development, pharma and biotech. My name is Giles, and I'm here to take you through this episode. Today's episode is taken from our winter 2018-19 issue, and is titled Beyond Send, Leveraging Non-Clinical Data to Drive Translational Research Forward. The article is written by Catherine Briggs et al. So now on to the main article. Beyond Send, Leveraging Non-Clinical Data to Drive Translational Research Forward. This episode will discuss some of the potential use cases that Standard for Exchange of Nonclinical Data, SEND, datasets can support, both to advance translational research across multiple research sites, and also to drive efficiencies with research partners, including contract research organizations, CROs. In addition, the authors of this episode will explore applications of consolidated, precompetitive data sharing to support cross-industry safety and toxicology applications. The Pistoia Alliance brought together a cross-functional life science industry group to review common challenges and opportunities in non-clinical drug development. The group focused their discussion on the challenges and opportunities for leveraging non-clinical datasets in support of translational research, and this episode provides a summary of their opinions and conclusions. The Standard for Exchange of Nonclinical Data, SEND, is one of the required standards for data submission to the FDA and specifies a way to collect and present non-clinical data in a consistent format. For the industry, standardization on the SEND format offers an opportunity to move beyond submission readiness to extract significant scientific and operational insights from the data. Increasingly, organizations are recognizing the potential of leveraging this high-value information for improving research operations both internally and with partners via data mining, visualizations, and AI and advanced analytics. SEND as a regulatory data submission format. The Standard for Exchange of Nonclinical Data Implementation Guide, SENDIG, was developed by the Clinical Data Interchange Standards Consortium, CDISC. SENDIG is intended to provide guidance for the organization, structure, and format of non-clinical tabulation datasets intended for regulatory submission and for exchange between organizations. Pharmaceutical companies and CROs are now required to submit SEND datasets for certain types of studies when filing investigational new drugs, INDs, and new drug applications, NDAs. SENDIG sets the standard for non-clinical dataset files containing electronic records of protocol design, animal demographics, animal exposure, and animal observation data, and detailing their content and terminology. SENDIG version 3.0 applies to single-dose and repeat-dose general toxicology and carcinogenicity studies. SENDIG version 3.1 adds standards for the electronic data records for certain types of safety pharmacology studies, and SENDIG developmental and reproductive toxicology, DART version 1.1, adds standards for embryo-fetal developmental toxicity studies. By creating a standard that is now required for submission of data, CDISC SEND is streamlining the communication between CROs, sponsors, and FDA during the process of study conduct, as well as at time of submission. CROs are increasingly using these formats to send interim datasets to sponsors. Sponsors can review preliminary data from their studies and perform early analysis of the results. More and more, sponsors are looking to CROs to provide SEND-compliant datasets to simplify the submission process. For CROs, implementing the systems and processes to produce SEND-compliant datasets can provide an important differentiator in a highly competitive market. Looking beyond the benefits of a common data model and its ability to provide the required datasets to regulatory agencies in the required format, SEND presents an important opportunity for biopharmaceutical R&D. As data complexity increases and the need for new therapies to meet unmet medical needs continues, SEND datasets, if leveraged properly, have the potential to provide unique operational and R&D insights. Such insights can increase efficiencies, reduce failure rates, and improve safety outcomes in the drug development lifecycle. Considering that one drug can cost an organization nearly $3 billion to develop to market, the leveraging of send data is an important means to increasing the return on an existing investment. Send and data mining, visualization and advanced analytics. Example 1. Operational metrics. Operational metrics are used by a laboratory to gauge the efficiency of its operations over time. This visualization relies upon the count of records in the lab domain over time, grouped by instrument name and instrument location. A convention of using the LB method raw data to include the instrument name and instrument location makes this possible. A stacked bar chart is then used to show the distribution of the work among different sites or laboratories where the analytical instruments exist. This can be useful to determine over time the workload taken on by each site within the laboratory network. The y-axis represents the number of tests done in that time period. The time period can be set by the visualization tool in blocks of different sizes, and a particular time period can be zoomed into in order to be specific to that time period in the analysis. You can also group the same information by instrument. This tells us which type of instrument is responsible for the bulk of the analysis during time periods. A trend over time provides information on which additional instruments may be needed to anticipate capacity issues. The visualization tools allow one to set the size of the date periods in order to look at a grosser or finer distribution of the data over time. The coloring is set to show where the maximum and minimum usages are occurring. The visualization tool allows filtering by other fields that are available in the send data. For example, filtering by study type and species to ascertain the relationship of the number of tests performed by location and instrument for each type of species and/or study type. Such operational information is the most useful for CROs which are doing high volume work and need to plan their equipment resources to align with incoming studies. Biopharmaceutical companies that are performing in-house studies would also benefit from metrics on the laboratory's throughput. Companies that outsource most or all of their studies could gain insight that would help them in future CRO selection and study monitoring. In the past, data on laboratory metrics may only have been available for individual studies. Now, a data warehouse of send datasets opens up the possibility to gain an overall view of the data history for many studies over time. This allows one to look for trends in types of analyses and where they are being carried out. This knowledge can be used to impact the cost and timeline for future study completion. Example 2. Data mining. In research, it is useful to have the flexibility to examine the data in an open-ended fashion, searching for patterns and correlations. The ability to review any of the in-life, necropsy, and histopathology observations across one or multiple studies is another benefit brought about by the standard representation of the data through send datasets. In this visualization, clinical pathology, organ weights, and micropathology data are available for filtering and exploring to search for meaningful correlations. Each section is colored or separated by dose group. The controlled terminology standards being developed for neoplasm and non-neoplasm allow cross-study comparison that was previously difficult due to differing terms as well as different ways to combine base terminology with modifiers. A heat map is just one way to see these dimensions. Stacked bar graphs show the data separated horizontally or vertically, which may be easier for some pathologists to see patterns that need further exploring. And scatter diagrams are often easier to see clusters of information from individual observations. Sunburst plots are another way of representing the incidence data as a series of concentric arcs, where one can zoom in or zoom out on the data represented from body systems, to tissues, to findings. Some may prefer to see this data represented as numbers in a table, with the added benefit of conditional coloring to draw one's eye to incidences higher or lower than the norm. Calculating correlation statistics is another ability of such tools. For example, clinical pathology data can be correlated against organ weight data to help the scientists concentrate analysis on areas of high correlation. All of these visualization types are being explored as pathologists are now having such data readily made available to them. The goal is to empower the pathologists to take full advantage of the standardized data across studies, compounds, and animal models in order to find toxicologic effects or candidate treatments for disease targets. Example 3. Study monitoring. Study monitoring and study analysis can also be enhanced by the ability of send datasets to be used with a visualization tool. The standardized data types and terms and the ability to receive in a dataset all the measurement types on a study make possible the development of tools to view the data in ways that the study director finds most conducive to their analysis. Clinical pathology is displayed as a scatter diagram, with time as the x-axis, the study visit day, i.e. the scheduled study day is used for time, and the test value as the y-axis. The clinical observation data is shown as a sparkline, with time along the x-axis and incidence as the y-axis. Each sparkline is for a different clinical observation. The body weight data is also shown as a scatter diagram, with time as the x-axis using the same timescale as the clinical pathology data. The y-axis is the body weight value. Color is used to show the different dose groups. The micropathology data is shown as a heat map. Each section down the graph represents a different dose group. The map is categorized by tissue and then the standard finding terminology. The charts are linked in that selections on one graph change the data displayed in the other graphs. This is done so that a selection of clinical pathology data points of interest will then filter the clinical observations, body weights and micropathology findings to those same animals. If the lab performing the study has the ability to deliver interim send datasets, the sponsor can view the data as the study progresses. This can be useful in looking for test article related effects or adverse events that might show up early in the study if the doses are too high, allowing for an early remediation if needed during the study conduct. Example 4. Advanced analytics and predictive modeling. A variety of opportunities to perform more advanced analytics and predictive modeling emerge if the challenges of study formatting and data capture into large databases are overcome. These opportunities range from implementing traditional statistical methods in an automated manner to more sophisticated data-led and machine learning techniques. From a single study perspective, novel implementations could include automated methods to surface anomalous findings, generating dose-response models for anomalous findings, and fitting simple pharmacokinetic and pharmacodynamic models to study data. When multiple studies are available, then emphasis can shift to either analysis of multiple compounds against any given study endpoint, or to a study endpoint specific focus, where the behavior of endpoints can be investigated. Hierarchical dose-response models that fit endpoint-specific data for multiple compounds in a single model and allow subsequent ordering of potency have been developed, but are not yet available commercially. In addition, endpoint-specific analysis that reveals, for instance, the background incidence rates of histopathology findings or putative maximum physiological responses for laboratory tests have already been performed on large datasets of preclinical data, but not yet on data formatted in the SEND standard. With the emergence of these approaches, enabled by adoption of SEND as an exchange format, the field will move towards increasing automation and more data-led toxicological predictions. Since data will be stored consistently in databases, it will become easier to generate data representations for any given compound that link compound with target, primary and secondary, and with perturbed preclinical endpoints. This enables toxicology to engage with system biology approaches that offer another approach to the prediction of clinical adverse events. Barriers to advanced analytics. The tools are available to leverage cloud resources to share standard formatted data across research sites and partners. However, challenges remain in leveraging SEND datasets to their full capacity. Sponsors may lack the infrastructure, e.g. resources and budget, to maintain a database and visualization tools. Also, instituting that infrastructure often requires staff to change business processes to connect data science with operations, particularly when moving from fragmented, locally owned data sources to centralized databases that offer richer data but more complexity. As a result, shared data that is available may be overanalyzed or even misinterpreted. In addition, there are key data omissions from the SEND standard that will limit its utility in terms of advanced analytics. For instance, there is no means to capture compound structure within the standard. In order to develop quantitative structure-activity relationship QSAR-based models from the datasets, compound structure data must be linked with a send data warehouse. Furthermore, there remain issues with the homogeneity by which information is captured in the standard by different partners. There is arguably too great a flexibility in how the representation of study design, dose group identification, and study elements are captured in the format. However, these issues could be overcome with a focused effort by stakeholders. Pre-competitive send use cases for safety tox. Widespread use of the send standard will facilitate pre-competitive collaborations by providing a standard format for sharing preclinical data and reducing the amount of harmonization needed in order to create integrated datasets. The Innovative Medicines Initiative (IMI) project, Enhancing Translational Safety Assessment through Integrative Knowledge Management (eTransSafe), aims to develop just such a dataset, utilizing data from 12 pharmaceutical companies for translational safety analysis. In particular, it plans to address the following questions. What is the relevance of preclinical safety studies for clinical study safety assessment? Can we refine preclinical experiments to better predict clinical safety? Can we build predictive models of safety outcomes from the data? By supplementing the send data with information on the structure and pharmacology of the drug, the dataset will support translational research analytics. As the amount of data in send format evolves, and by combining it with legacy data in the same format, it will be possible to identify the background incidence of histopathological findings, along with the most common drug-related pathologies, and determine reference ranges for clinical pathology. Drilling down further into this data can identify the impact of study duration, species specificity, and gender differences on the findings observed. Conclusion. Sponsors are now required to submit data from preclinical toxicology studies started after December 17, 2016 to the FDA in send format for investigational new drug (IND) applications and new drug applications (NDAs). The send format creates a structured terminology and consistency for regulatory submissions and allows for concise communication between the FDA, CROs, and sponsors. Moving beyond the objective of regulatory compliance, the send format standardization represents a significant opportunity for drug developers to extract valuable scientific and operational insights from preclinical data. From enhanced study monitoring and visualization of operational metrics to reducing drug failure rates and improving safety outcomes, the potential is significant at a time when risk and complexity of drug development continues to rise. Eventually, these datasets can be leveraged to build predictive models that reduce the burden of in vivo testing and help identify and validate novel targets. Ultimately, these efforts will complement systems biology approaches and serve to improve the drug development process, leading to better medicines being available for patients in a timelier fashion. This article was written by Catherine Briggs, Bob Friedman, Kristin Ferrara, Mark Pinches, Will Drew, Cheryl Reel, Melody Thompson, Antoinette Hayes, Jason Gratt, Shyla Wiley, and Mary Beth Walsh. Catherine Briggs is Research Leader at Alhazza Limited. Bob Friedman is Chief Technologist at Zybion Medical Systems. Kristin Ferrara is Director, Global R&D IT Programs at Takeda Pharmaceuticals. Mark Pinches is Senior Principal Scientist at Alhazza Limited. Will Drew is Principal Global Alliance Manager at Alhazza Limited. Cheryl Reel is Head Non-Clinical Writing and Document Management at Alnylam Pharmaceuticals. Melody Thompson is Manager Non-Clinical Writing at Alnylam Pharmaceuticals. Antoinette Hayes is Lead Associate Scientist Toxicology Study Manager at Alnylam Pharmaceuticals. Jason Gratt is Principal Software Engineer at Takeda, formerly Millennium, Pharmaceuticals. Shyla Wiley is Manager Drug Safety Research and Evaluation at Takeda Pharmaceuticals. And finally, Mary Beth Walsh is a consultant with the Pistoia Alliance. If you've enjoyed this episode, then you can subscribe to Drug Discovery World free of charge by visiting our website at ddw-online.com, where you can also view all of our articles, including references and images, and download the original PDFs. You can find the links in the show notes. If you're enjoying the podcast, then do leave us a review and subscribe, and you can also follow us on LinkedIn and Twitter for updates. Thank you for listening, and we'll hope to see you in our next episode.